Okay, if not, we will invite next speaker, Dr. Christopher Grupen. He's from the University of Sydney. He graduated Adelaide University in 1986 and had a position researcher as a research assistant in Vitro Limited, the private company for the technology. And he got PhD under the supervision by Dr. Hiroshi Nagashima at Meiji University, Japan. After having PhD, he moved to the University of Adelaide as a postdoctoral researcher. And from 2006, he has been a senior researcher at the University of Sydney. So today's presentation title is Current Status of Porcine Embryo in Vitro Production. So he's very concentrated to the embryo production, porcine embryo production in vitro. So his paper was invited to the Cellulogenology last year. It is the anniversary issue, it is 50, volume 50. So he presented a very nice paper in Cellulogenology. Also, this presentation, please check his review paper at home. Please.
Thank you very much for that kind introduction. Firstly, I'd like to start by thanking the organisers for giving me the opportunity to present at this fantastic satellite symposium. And of course, I'd also like to acknowledge the fantastic hospitality so far, and I really look forward to the conference dinner tonight and maybe a little bit more karaoke. And I'd like to particularly thank Dr. Nagai for asking me to talk about this very important technology. I was very honoured when he asked me to do so, and then after I accepted, I realised what I'd gotten myself into and thought, wow, this is a little bit overwhelming. It's such a large area, it's incredibly difficult to do it justice in a half-hour presentation. And I wondered how I was going to go about it. And I thought, well, I'm going to present to you today a general overview of the area to give perhaps those in the audience that aren't familiar with the area a better understanding of where this technology currently stands. And perhaps those in the audience who are familiar with the area, there's not so much new information, but hopefully towards the end I'll present some of my ideas that we can discuss and about what I think is the future of the technology and where we're going. Just as a starting point, really the significance of being able to produce embryos in vitro really began about trying to understand basic gamete and embryo physiology, and that research still continues today. It's very, very important. We still don't know everything there is to know about the biology of the embryo. And of course, initially it was seen as a hugely appealing technology because perhaps we could utilise the thousands of oocytes in the ovaries that are just normally wasted. Certainly in monovular species, cattle and sheep, that has come to fruition. In the pig, it hasn't really been realised fully yet, but still there is that potential to use many more of the female's oocytes and disseminate valuable genetics. Of course, producing embryos in vitro, it can be used as a tool just to assess the fertilising capacity of sperm from different males. And of course, generating embryos in vitro in the lab, many groups do this to then develop associated reproductive technologies such as somatic cell nuclear transfer, so cloning technologies, or to improve embryo cryopreservation procedures. So embryo in vitro production is very important for a number of reasons, but I do want to emphasise that really the driving force for all of the developments really over the last 10 to 15 years has been to generate large numbers of embryos to be able to produce genetically engineered animals for agricultural and biomedical applications. Really, the push to produce genetically engineered or genetically modified pigs has been the driving force for many of these developments. And just to emphasise that point, I've just got a list of some of the genetic modifications that have been achieved over the last 15 years, 10 years in pigs. I prefer the term genetically engineered rather than genetically modified. Bruce Whitelaw from the Roslin Institute, I saw one of his presentations a few months ago, and he convinced me that it's much better to say genetically engineered because it implies that it's a much more precise manipulation of the genome. And just the list here that I have here is really a snapshot. Probably the most famous GM pig is the Enviro pig for agricultural applications. Vitae is produced in the salivary gland. Of course, the potential benefits to production systems would be huge if we could get approval for these animals, but sadly we don't have those approvals yet. And of course, this dramatically reduces phosphate in the feces, something like 70%, so it would be massively beneficial to use these animals in production. Pigs that have alpha-lactalbumin produced in the milk would dramatically improve piglet survival. And very recently, researchers at the Roslin Institute, they announced the birth of Pig 26, which is really a major breakthrough and probably the start of a new revolution, using very, very specific gene editing tools. They introduced a point mutation into the genome and conveyed resistance to African swine fever in domestic pigs. So this really promises to be perhaps the future of where this technology is heading in terms of generating GPs, very precise gene editing tools. Of course, biomedical applications really have been prominent in terms of generating genetically engineered pigs over the last 10 years. Of course, the xenotransplantation studies, the benefits for using pigs as organ donors is self-evident. And of course, this ever-growing list in the last 10 years, many more models, pig models for studying human diseases have been generated. So I just want to emphasise the fact that really this technology is the driving force for improving embryo production systems. Now, in terms of the holy grail for reproductive biologists in my area, the holy grail has been how do we produce the best quality embryos? And of course the equation is pretty straightforward. If we have good starting material, if we can select the best quality immature oocytes and provide the optimal conditions for in vitro maturation, in vitro fertilization, and embryo in vitro culture, then we'll achieve that. And that's really what all of the studies in the area are hoping to achieve. And so I've broken up my talk into these 4 areas. And because of the limited time, I'll be flying through each of those areas. In terms of the factors affecting oocyte quality, we've known for a long time these basic factors that influence quality are maternal age, so oocytes from prepupal animals are of lower quality than those from adult animals. Of course, follicle diameter affects oocyte quality. The number of cumulus cell layers affect oocyte quality. Just some data from a study from my lab a few years ago now. We looked at oocytes collected from adult animals and prepupal animals We recovered the oocytes from different sized follicles, small, medium and large. We then fertilised them after maturation and looked at their ability to form blastocysts in vitro, so that's a measure of oocyte quality. You can see that from the adult animals, we consistently got 50 to 55% blastocyst rates from all of the follicle size classes. Prepupal oocytes, however, only those from the large follicles were good quality, medium was significantly lower, and small oocytes from small follicles had quite poor development. Now the point I want to make on this graph is that this group here, this is really the population of oocytes that we're dealing with mainly when we're trying to generate embryos in vitro, because from slaughterhouse ovaries, market dictates that pigs are slaughtered when they're prepubal, and on prepubal ovaries, the vast majority of follicles are small. There are very, very few large antral follicles on the prepubal ovaries. So really the challenge is we've got to improve the quality of this group of oocytes. Other work in my group, we've shown that season affects oocyte quality as well. So some of the period of seasonal infertility has a negative impact on oocyte quality. And you can see that in this graph where we took oocytes from small and large follicles, and over several years we collected in summer months and winter months, which in Australia the seasonal infertility period is February-March. And you can see that here in the large follicles, small follicles, there was no difference between summer and winter. But in the large follicles, again, this is blastocyst formation rate. In winter, we were getting the 55% blastocyst rate, but in summer, that was dramatically reduced down to the poor quality of oocytes from small follicles. So you've also got to consider when you're collecting your ovaries. Indicators of oocyte quality, I just have a list there. We've known for a long time factors within the oocyte, such as glutathione, are critical for successful fertilization and pronuclear formation. We know that oocytes from small follicles have a poor ability to accumulate cyclic AMP, the key regulator of meiotic progression. Oocyte secreted factors are important as indicators of oocyte quality. Of course, with some of the new techniques now, transcriptomic analysis, some transcripts have been associated with oocyte quality, so differences between oocytes from small and large follicles. Follicular fluid, the environment that the oocytes are bathed in, that has been examined extensively in numerous studies, and of course steroid levels increase dramatically as follicle size increases, so there's a correlation there. And now metabolomic analyses are allowing us to understand what the oocyte, what cumulus cells within the follicle are utilising. So amino acids and intermediates of the tricarboxylic acid cycle change as the follicle increases in size. So we're understanding that there are shifts in energy generation mechanisms. In terms of the optimal oocyte in vitro maturation systems, very, very briefly, it's incredibly important to synchronise nuclear maturation because of this premature meiotic progression in vitro. So a critical aspect of these systems is to elevate cyclic AMP levels within the oocyte. There are a number of methods of doing this, a number of different treatments. And really the only treatment that has been shown to be consistently beneficial is a cyclic AMP analog called difuryl cyclic AMP. There are other treatments, but findings between studies are that consistent. In terms of improving cytoplasmic maturation, of course, you can throw the kitchen sink at the oocytes to try and improve their quality, so try and throw in as many growth factors as possible. Follicular fluid, undefined composition, but clearly there are very important factors in follicular fluid that improve cytoplasmic maturation. Of course, hormones have been manipulated within the maturation system to improve quality of the oocytes. And one of the early treatments that was shown to improve oocyte quality were agents that maintain high levels of glutathione within the oocyte. So antioxidants to protect. I've got a list there of growth factors that have all been shown to provide some improvement in terms of cytoplasmic maturation. Sorry, I don't have time to go into details on each of those, but I will very briefly mention follicular fluid. I just said that it's undefined, so we don't know exactly what components are having positive effects, but we do know that they provide a protective, very, very important protective effect during maturation and reduce cumulus cell death or apoptosis. This figure here, the top three panels are one cumulus oocyte complex. The green, I'm not sure whether that's showing up very well, but they're apoptotic cells. The red is total cells, and that's the merged image. So the top three is one COC, the bottom three panels are one COC. And the top three are, this is high levels of apoptosis versus low levels of apoptosis. In our experiment with follicular fluid, we looked at changing halfway through. In the first part of maturation, we got significant reduction in apoptosis with follicular fluid. But the really important part came in the second half of maturation. So if there was no follicular fluid present for the entire maturation interval, we had very, very high rates of apoptosis, including it for the entire time, very, very low rates of apoptosis. So clearly there are factors in follicular fluid that are important. But interestingly, the group that had follicular fluid present just for the second half also had low apoptosis. So it seems that the second half of maturation is the critical period where you've really got to look after the cumulus cells. The flip side of that is that the protective effect also has a stimulatory effect, and this was seen in increased steroidogenic activity of the cumulus cells. We looked at progesterone concentration in the spent media after maturation, and this graph, these are the same groups as was shown on the previous slide, same time points, but the graph is virtually a mirror image or a reverse image of the previous results. So in other words, the groups that had the highest rates of apoptosis also had the lowest progesterone levels, and the groups that had the lowest rates of apoptosis produced the highest progesterone in the maturation media. So clearly, looking after the cumulus cells maintains their functionality, and that they can actually produce the progesterone that they should. And that correlated nicely with blastocyst development. So the three groups here that had some follicular fluid for maturation or for the entire period, they had good rates of blastocyst formation, around 45%. The group that didn't have any follicular fluid had the poorest rate of blastocyst formation. And the interesting point here was that this level of progesterone, around 800 to 1,000 nanomole, correlates very nicely with the levels seen in the follicle. So we propose that there's a threshold of progesterone that needs to be there for good oocyte quality. Very briefly, maturation media that have been used, really the two main ones, the mid-90s, North Carolina State University-based media was described, and more recently, porcine oocyte maturation medium, POM. The main differences are the carbohydrate composition and the amino acid mixtures. But the point that I want to make on this slide is that this media has been a very, very important advance because it's still highly effective when you replace the follicular fluid with polyvinyl alcohol. So you can work with a fully defined, chemically defined maturation media. And this is really being important for the area to make further advances where we can understand specific additions. In vitro fertilization, another whole area that I could talk easily half an hour on, but I only have a few minutes. The problem that really still remains in pig IVF is the polyspermic problem. We still have that issue. And in vitro matured oocytes have a reduced ability to provide this blocked polysperm. We know that there's an effect of maternal age. So oocytes from prepubertal animals have higher rates of polyspermic fertilization than those from adult oocytes. And clearly the insemination conditions affect polyspermic rate. So numbers of sperm per oocyte and the duration of gamete co-incubation. However, it's not just an easy matter of reducing the number of sperm in the insemination droplets, because if you do that, you also get very low fertilization rates. And that's always been the problem. We want to keep high fertilization rates and just reduce polyspermy. And still, we cannot achieve that. I just want to mention briefly that the other issue with polyspermic fertilization rate, it's not that you're just producing an abnormal embryo, but then those abnormal embryos are still actually able to grow in your culture system and form blastocysts. So you get either triploid, aneuploid, or mixoploid embryos. And in fact, these mixoploid embryos, only about 10% of polyploid embryos, can actually form offspring. So using blastocyst formation rate is not really a very good indicator of your viable embryo production rate. You can centrifuge the fertilized oocytes, polarize the lipid, look at the pronuclei, and determine whether it's a normal diploid zygote. These can be put into recipient animals and generate offspring, so there's no detrimental effect of this centrifugation. But of course, it's another step and it's more labor intensive. A whole lot of studies, many studies have been done to try and reduce the polyspermic fertilization. So trying to mimic the in vivo conditions more closely by adding oviductal factors to the IVF conditions. An interesting strategy has actually been to try and harden the zona pellucida so it makes the zona more resistant to sperm penetration. That has reduced polyspermic rates marginally, and a whole host of methods have been developed to try and basically act as an obstacle course for the sperm, so only the best of the best sperm actually get to the oocytes and fertilise them. And I don't have time to describe each of these systems, but they really are all just trying to provide some sort of barrier so that only the best sperm reach the oocytes. I'll very briefly just describe a method that we use in our lab. We've been using it for a long time now. It's a very simple modification of IVF. Conventional IVF, you just simply throw the sperm and the oocytes in the insemination droplet together for 5 or 6 hours. With the simple two-step IVF method, we just co-incubate the gametes for only 10 minutes. And amazingly, that's all the time that you need to get the first sperm to bind loosely to the oocytes. Then you very carefully pick out those oocytes and loosely bound sperm, transfer them to fresh insemination droplets. And interestingly, you actually improve fertilization rate. We found that we got higher rates of fertilization doing that. We reduced the number of penetrating sperm per oocyte, and we actually improved embryo development rate, cleavage and blastocyst formation. So we feel that it's important to remove the oocytes with the loosely bound sperm to fresh insemination droplets so that you're taking them away from those excessive numbers of sperm in the insemination drop, which then undergo cell death and are providing detrimental impacts to the oocytes that remain. Other researchers have utilised the two-step method, and they've found that the effectiveness of the method does differ between bores. So of course you do have to tweak the system for each bore, and of course it depends on the sperm-oocyte ratio. So one strategy that's being suggested is to combine methods. So combining the two-step method with a straw IVF system. They got good fertilization rates. However, they still obtain polyspermic rates of around 23 to 42%. So we still haven't been able to overcome it, but perhaps this strategy of combining selection methods is something that can be explored a bit further. Pig embryo in vitro culture. This image is very kindly provided by Dr. Takashi. Thank you very much. I just have a list of some of the media that have been used to culture embryos, pig embryos, which then have been transferred to recipients and live offspring have been generated. And this is an important picture because these are the first piglets that were produced from in vitro produced embryos that were then cultured to day 5 or 6 in vitro, then transferred to recipient females and generate live offspring. And this was achieved in 2002. It was actually achieved using NCSU-based media or modified NCSU. And now, having said that, I'm now going to say that in subsequent studies, in comparisons, numerous comparisons, have indicated that porcine zygote media, described by Yoshioka, is the superior medium. Its composition is based on porcine oviductal fluid components. It's supplemented with amino acids. And again, the important point about this media is that it is effective when supplemented with PPA, so it's fully chemically defined. Really just a basic point that composition of the embryo culture media is really trying to meet the requirements of the developing embryo. Amino acid supplementation has been found to be very important. However, high levels of amino acid can be detrimental because of the build-up of ammonia. Recent studies have shown more stable forms of amino acids are beneficial, and an interesting finding was that glycine acts synergistically with glucose, so when they're both added at around day 5 to 7, they improved embryo quality. A long list of antioxidants and growth factors have been added to embryo culture media for the pig, and all of these have been found to show some benefit to development, whether it's blastocyst formation rate or blastocyst cell number. So many studies in this area as well. This slide I probably should show for a little bit longer than I will, but it just gives you an idea of how many milestones have been achieved in pig embryo IVF over the last 10 years. Really, in terms of using chemically defined media and generating, so all of these studies generated piglets, and all of these are in vitro produced embryos, either by IVF or through somatic cell nuclear transfer or cloning. We've also been able to vitrify these embryos, thaw them, transfer them, and get piglets. So many milestones have been achieved, and in research settings, it really has been an exciting time over the last 10 years to see how much things have improved. Summary, I've discussed this already, so I'll just, in the interest of time, flick through to research directions. I feel that a very important area is to manipulate the quality of the oocytes before we even start using maternity nutrition, and of course that's not only important for in vitro production, but of course just animal production through normal mating. So this is going to be a very important research area. In terms of improving the IVM systems, of course these also secreted growth factors that are being looked at in more detail. EGF-like peptides, I think they're going to really make a difference in the coming years. Fatty acid metabolism, we still don't understand why the pig oocyte has such a high amount of cytoplasmic lipid droplets. And I've just got this slide up here to show some of the technology that's used in the human IVF setting. We can now use polarized microscopy, microscope called the pulse scope, to actually look at how well the genetic spindle is organized. So this, I think, will be a very useful tool to non-invasively pick out the oocytes that are very best quality. In terms of IVF, I would suggest that perhaps combining sperm selection methods and of course looking at in more detail individual boars, what their differences are and what we need to do to optimise the conditions for each. Briefly, I have a study here that, or certainly understanding the proteins involved in the sperm-oocyte interaction is going to be critical, and certainly with proteomics we can do that in a lot more detail. Just a few weeks ago, Bianchi, I think the group in Cambridge, they identified Juno, the receptor for the Zumo protein that's on the sperm membrane. And so this study was done in mouse, and I think this is going to be very, very important, looking at the receptor for this sperm protein, critical for the interaction. When they blocked Juno, they blocked fertilization. So I know this is going to be a very interesting research area. In terms of in vitro culture, there are going to continue to be improvements, trying to better meet the requirements of the developing embryo. I've got a picture up here of the embryo scope. Again, this is in the human IVF setting where you can track, do time-lapse observations of individual embryos. So a very, very powerful tool to understand the differences between embryos in terms of the kinetics of development and their growth. So really, there are a whole lot of advances that are probably technology-based that I think will improve in vitro production systems. I have to mention the associated technologies are equally important for using IVP pig embryos, and I think the next two speakers will clarify that more. So, of course, we need more robust cryopreservation systems, and the issue is to be able to restore very large numbers of pig embryos. And the actual embryo transfer method itself, that needs to be improved. Cloning is still the driving force. I mentioned at the beginning about the gene editing. I think that will be the new driving force because we won't need to generate somatic cell nuclear transfer embryos. We just do the manipulation directly on the zygote. So no more cloning needed. Just generate really good quality embryos. And who knows, with all of these improvements, maybe in the foreseeable future, these technologies will come together and be efficient enough to be a commercial reality, to generate in vitro produced embryos for advanced breeding schemes, probably more realistically to save or conserve genetics of rare and endangered breeds. And I'd like to finish on that note. Thank you very much.
Thank you very much. So, do you have any simple questions or comment on this presentation? Okay, if not, please.
Okay, thank you also for a very extensive review, Dr. Cooper. Well, I was particularly called by your statement regarding we still don't know everything about this complex biotechnology. Well, does this mean that shall we stop tinkering with nature and subscribe to the divine creation theory that we go the natural way of reproduction? But if ever it's not the case, then can we have our acts together? You mentioned about the prepubertal oocyte characterization. So there are intervening protocols towards full follicular maturation or successful fertilization. So how about the sort of identifying what appropriate physical character of the prepubertal oocyte that you will identify that would warrant that when it will be suited with the proper intervening protocols from prepubertal to full maturation, and it will match because of that characteristic of the oocyte, then I think this will be an appropriate research direction for everybody to be involved. Can you care to comment on that?
Well, there is still so much that we can learn, and certainly I feel that with the advances in technology in recent years, we are going to continue to gain more technology. When you look at the transcriptomic information, the proteomic information, the metabolomic information, each of these have been done in isolated studies. If we can draw that information together and get a better, fuller picture of what is happening in the follicle, the other really important aspect to this is that the technologies have advanced to the point where we can analyse individual oocytes and individual embryos, and that's been the limiting factor up to now. We've always had to work with groups of oocytes or groups of embryos, and of course there are variations across the group, but we haven't been able to get enough material to do the proteomic analysis or the transcriptomic analysis. But when we can do it on individual oocytes and then follow that through in the individual embryo, that's when massive improvements will be made.
Agreed. Another comment? Question? Okay. We have a lunch break until, please give the information.
Okay, thank you very much. Another big hand to Dr. Grupen. And of course, our moderator, Dr. Kikuchi. We will pause for our lunch break. Please be back at 1:40. We're following that wall clock. Okay, so we will be back at 1:40 for speaker 5, still on session 2, oocyte and embryo factory. All right, thank you, and we'll see you at 1:40.